I was going to give you 50%, but now they said 20. That sounds better.
I take that.
All right, it looks like it's 9 o'clock now, so we can get started. So please spread out. So that way there's at least a chair between each of you. And please put everything away except for a pen or a pencil or both if you want. All right, so please keep these face down until it's time to begin. And then, again, just a reminder, once you are finished, please hand them to Dr. Cato or myself. And you are welcome to return to your seat and just sit back and relax, or you may exit the room.
Discuss everything we got wrong.
But not too loudly.
Does anyone not have a quiz? Okay, please begin.
minutes. One more minute. One more minute. Okay, time is up. Please pass your quizzes to the aisles. All right, I'm assuming you'll take a shot at those. All right, thank you. All right, I'll send them in. Thank you. All right, come on in.
No, that was the one, but it wants to come here.
But I understand, but you use two, right? Then you make it.
Yeah, yes, yes. Right.
No, that was fun.
Glad you liked it. Great. All right. So let's get started. We have today our last lecture talking about sugars. So this pathway is something that kind of melds a lot of the things that we've already talked about together in a new and different way. And so it's a nice way to end our talk about sugars in something that kind of brings a lot of it back together. So this is a pathway called the pentose phosphate pathway. And a lot of times you just see it abbreviated as PPP, or you could say triple P, whatever you like to say. Or if you just want to say it all at once, pentose phosphate pathway, that's good too. All up to you. So we're going to talk about what happens during the pentose phosphate pathway, what goes in, what comes out, and really we're only going to be concerned with a couple, the rate-limiting enzyme and the types of enzymes, not the actual names of them. Then we're going to describe what happens during phase 2. So as you can see, phase 1, phase 2, 2 phases. And then we're going to relate how the triple P pathway is used to base off the cell's energy requirements or its need for nucleic acids. So it kind of links sugar and nucleic acid syntheses together. And then we're going to describe how we use this pathway to help us define against reactive oxygen species. So with our human metabolism pathway, we've covered everything now in blue. And today we're going to talk about this area here in red. Technically, you've already done all of this here with DNA and RNA with Dr. Domina. And so you can see that this is a merge between the sugar pathway and the nucleic acid pathways. So in going over this pathway, we're going to talk about it in terms of a clinical case. So we have a 32-year-old male admitted to the emergency room in a semi-comatose state. Early in the morning, he was listless, anorexic. His skin appeared yellow, as well as his sclera, the whites of his eye now look yellow. So a bit jaundiced. He complained of abdominal pain. He passed very dark urine and had vomited several times. His physician, of course, was like, this is not normal. You should go to the emergency room. And so he had no foreign travel history. He does not use herbal medications or illicit drugs. He had blood drawn, and the lab values were shown here. So his hemoglobin is very low. His platelet count and his white blood cell count are normal. So only his red blood cells are doing poorly. His white blood cells and the platelets are doing fine. And the bilirubin is also pretty high. Bilirubin is something that's made when you break down hemoglobin. So we have low hemoglobin, high bilirubin. The two values match. Yes. No, at this point, this is all we have for values. So from what we can see is he's showing some signs of anemia. So his just not getting enough oxygen through that hemoglobin. And so he's given a blood transfusion. So we'll leave him here like this, and we'll try and work back to what could possibly be going wrong with him. So let's just give an overview of the pentose phosphate pathway. So this is a pathway that, as we said earlier, merges some of the sugar carbohydrate pathways with the nucleic acid pathways. It occurs primarily in the cytoplasm. The input is glucose 6-phosphate, and it occurs in two phases. The first phase is the oxidative phase. So we're going to produce 2 NADPHs. So this is very similar to NAD, except it's phosphorylated, but it's very important and different because enzymes that use NAD cannot use NADPH, and enzymes that use NADPH cannot use NADH. So there is a distinction, and it will also produce CO2. You're, of course, inputting that glucose 6-phosphate, which is a 6-carbon sugar, and you end up with a 5-carbon sugar. So you're only losing 1 carbon in that CO2. And then the second phase is the non-oxidative phase. So here we're just converting sugars. So we're going to either put them into making nucleotides, we're either going to make them into other molecules, or we're going to put them through glycolysis. A lot of options at that point. And so the pathway is usually regulated by the concentration of NADP+ in the cell. So high levels will promote this pathway, because when you're at NADP+, you want to regenerate the NADPH. In this pathway, no ATP is generated. So this is not something that we use to regulate our energy balance. So when we look at the pentose phosphate pathway and glycolysis, here we have glycolysis down the center, starting with glucose, ending with pyruvate. It's a little truncated, but you end up with pyruvate at the end. So glucose, the first step is getting that glucose 6-phosphate. When you want to do the pentose phosphate pathway, you can shunt this glucose 6-phosphate out of glycolysis and put it into the pentose phosphate pathway to make ribose phosphate. And you can also shunt it right back in at the fructose 6-phosphate stage and the glyceraldehyde 3-phosphate phase. So here the pentose phosphate 2 phases are color-coded. The blue is the oxidative branch, where you're producing a 5-carbon sugar, which is ribose. And then the black bars is that non-oxidative phase, where we're converting sugars, where if we don't need ribose, we can turn it over into fructose 6-phosphate or glyceraldehyde 3-phosphate. So this is just general overview. Now we'll look at what exactly happens in each of those phases, the oxidative and the non-oxidative. So first we'll start with phase 1, which this is the oxidative phase, where if you were to look at the pentose phosphate pathway in whole, it's only this little part here in the white. So the pentose phosphate pathway, the oxidative phase, the first phase, occurs in 3 steps, of which you're going to regenerate your NADPH twice, in the very first step and in the third step. You go through 2 oxidations, starting with your glucose 6-phosphate. You have an oxidation to a lactone, which is a cyclic ester. Then you break that cyclic molecule into a linear molecule. You create another NADPH, and you end up with ribulose phosphate. Now, the only enzyme here that's really important is this first one, which is the rate-limiting enzyme. This gets regulated, and this will let the whole pathway proceed. If this enzyme is not active, you're not going to get any of the phases of the pentose phosphate pathway. So this enzyme is glucose 6-phosphate dehydrogenase. So we start off with glucose 6-phosphate, and we end up with ribulose 5-phosphate. Now that we have ribulose 5-phosphate, we can just rearrange it using isomerase to make ribose 5-phosphate. So all we're doing is moving a carbonyl carbon, and we end up with ribose 5-phosphate. Now, ribose is something that we use very much in our cells, and we have many uses for it, one of which is nucleotide synthesis. So ribose 5-phosphate, using PRPP synthetase, can create PRPP. Now this should be reviewed from lectures earlier about purine synthesis and pyrimidine biosynthesis. So we're not going to go any further. Once we have this PRPP, you can go down and make all of your nucleotides and of course then use them to make DNA, RNA, what have you. And so the whole point of the very first phase of the pentose phosphate pathway is to take your glucose 6-phosphate and make ribose. And in doing so, you're going to regenerate that NADPH. That's the entire reason it exists, is to regenerate that NADPH. Yes?
So at the end of phase 1, is the final product ribose 5-phosphate or PRPP?
Ribose 5-phosphate. This will go further to make the PRPP only if you need the nucleotides. So that's one of the uses of actually the ribose 5-phosphate. And we'll talk about other ways you can use the ribose 5-phosphate other than using it in nucleotide synthesis.
Okay, that's dividing cells.
Yes, exactly. Looking ahead. So now we move on to phase 2 of the pentose phosphate pathway, where we're going to take that ribose 5-phosphate and not convert it into nucleotides, but instead convert it into different types of sugars. And all of these sugars are then going to feed into the glycolysis pathway. So our goal is to create a reversible link between pentose phosphate pathway and glycolysis. Essentially, you're taking out of glycolysis that glucose 6-phosphate, so it can't go anywhere through. And the whole purpose of that is to get that NADPH. Now, if you don't need nucleotides, you can then put it right back in, so that net, you're not really losing ATP by doing this reaction. You can put everything back in and still get energy from that starting glucose molecule. So we do this through cleavage of that ribose phosphate by 2 types of enzymes, either transketolases or transaldolases. Now there's specific names for them. Just be concerned about what types of enzymes there are. We're not going to talk about the specific names of them. Just a transketolase always cuts an alpha cleavage after carbonyl, And transaldolase will do a beta cleavage. And we'll look at that in the next 2 slides. So the transketolase, you always have a 2-carbon unit transfer. So it cuts its substrate and then adds 2 carbons onto another molecule. So here we have, for example, a ketose and an aldose. You have the transketolase coming in. It cuts off 2 of the carbons from that original ketose and adds onto the aldose 2 carbons as well. So now we have this 2-carbon unit transfer. So you see N, we have N minus 2 on the other side, M amount of carbons, and we have M plus 2 on the other side. So that's a transketolase. The transaldolase does something very similar, but instead of transferring 2 carbons, it transfers 3 carbons. So if you start off with N carbons, and then you add dihydroxyacetone phosphate, the aldolase will then add 3 carbons onto that aldose to create this ketose. So you can see there's a 3-carbon unit transfer. So ketolase, 2 carbons, aldolase, 3-carbon transfer. So I hope you can zoom in on this better in your slide. What type of reaction would be seen here with this type of reaction? Is it working upon by a transketolase or transaldolase? All right. Looks like it stopped moving. Everyone have a chance to answer? No. All right. Okay, everybody? Yep. Okay. So we have majority class saying transketolase, and then about a third of the class saying transaldolase. So it is a transketolase. So if we were to label how many carbons are in each of these molecules, we start off with a 4-carbon and a 5-carbon. At the end of the reaction, we get a 6-carbon and a 3-carbon. So total, 2 carbons were moved. So 2-carbon transfers are done by transketolases, whereas those transaldolases transfer 3 carbon units. So really with this, all you need to do is count how many carbons are moving. So how do we use these transketolases and transaldolases? We're just moving carbons around. What's the purpose of this? So in the non-oxidative phase, we will first convert our ribulose into the ribose. We've already done the ribulose into the ribose phosphate, and we can then do an epimerase. So remember when we talked about sugars, epimers are just different arrangements of these same sugars of the same length. We're just moving where those OH groups are. So we then produce a xylulose 6-phosphate instead, because we just moved where an OH group was with this epimerase. And then we do some transketolase reactions where we end up with a 5-carbon sugar and add it to another 5-carbon sugar. We get a 3-carbon and a 7-carbon sugar. But this 3-carbon sugar here is one that we are aware of and know that can enter glycolysis. We have a glyceraldehyde phosphate. So that's good. We can put that into glycolysis. This glyceraldehyde phosphate can also be combined with a 7-carbon sugar. You can make a fructose 6-phosphate. That also can go into glycolysis. That's great. And then you can also transfer some of these other ones, and all at the very end, you end up with making 2 fructose phosphates and 1 glyceraldehyde 3-phosphate. Now, all of these reactions and how they occur are not important. It really isn't. All I would like you to know is at the end of the non-oxidative phase, you get these three molecules, 2 fructose 6-phosphates, 1 glyceraldehyde 3-phosphate. And what do we do with them? We put them into glycolysis. We can then just feed it right back in, which is great because you aren't wasting any of that glucose 6-phosphate that you started with that could have gone through glycolysis and further down into TCA cycle and oxidative phosphorylation. You're shunting them right back in. You're not losing the power of that glucose by going through this pentose phosphate pathway. You're just going about it in a more scenic route rather than very direct. So, review question. So the pentose phosphate pathway produces blank as an intermediate that is used in the synthesis of blank.
What is a blank?
So the first one is the first blank, and then dot, dot, dot. The second one's the second blank.
What should be there?
Words that are possibly on there, A, B, or C.
Or D?
Or D. Yeah, maybe not. Maybe it's not D. It's not D. Let's just cross that one off right now.
I'm gonna pick D.
All right, Derek, go for it.
I believe in you.
I don't know.
That wasn't me.
I don't know, Derek. I think it was. All right, everyone have a chance to answer? All right, so no one picked D. Okay, that's good. Remember, this pathway does not produce ATP at all. So these no. So we have A, B, and C, all of which start off with something that is actually produced with the pentose phosphate pathway. We get fructose 6-phosphate, we get glyceraldehyde 3-phosphate, and we do get ribose 5-phosphate. Now, the key is, what do they actually make? Both choice A and choice B, we don't really use them to produce glycogen. You never are going to put a sugar in and get a protein out later, so it can't be B. What you get is you're putting ribose 5-phosphate in, and then you can get eventually DNA and RNA. You're making nucleotides. The nucleotides can be polymerized together, and eventually you get DNA and RNA. The ribose 5-phosphate is really just produced at the end of that first phase. The second phase is what's going to be producing those other two, the glyceraldehyde 3-phosphate and the fructose 6-phosphate. So now that we've become familiar with the pentose phosphate pathway, what does this have to do with our patient? So further investigating EC's condition, his doctor finds that he has hemolytic anemia, which basically means his red blood cells are bursting open. And so that's interesting in itself, and this is a picture of what it looks like. You have our normal red blood cells, and then you have these ones that are kind of like wonky looking, and eventually they burst. So in talking with the patient, the doctor finds out that EC occasionally feels sick after he eats certain foods. So naturally the doctor asks, what did you eat last night? And so the night before, his family ate fava beans, which they grew in their garden, which is something they hadn't done before, but they started growing them this year out of curiosity to try it. And so this sent off a little red light in this doctor's mind. So the blood tests reveal that EC has something called favism, or glucose 6-phosphate dehydrogenase deficiency. So basically, he doesn't have this enzyme that is working, so his pentose phosphate pathway isn't functioning properly. It's actually a very common condition. About 400 million people globally have this disease, so that's a very large chunk of the human population. And it's very common in people of Mediterranean descent, not just Mediterranean European descent, Mediterranean African, Mediterranean Middle Eastern. All around that, all around the sea, people have this at a very high percentage. And so there's varying presentations of it. Males get affected with it much more severely than females do. It's due to the genetics of it. It's X-linked, so males only have one copy, females have two, so there's a little bit of dilution of the effect in females. But question is, he has poor functioning pentose phosphate pathway. Why would this then lead to the hemolytic anemia? What is going on where ideally this pathway should be working in every single cell of his body? The enzyme is deficient. It doesn't work at all. And so technically that should be all over his entire body. Why is it only in his blood cells that there is a problem? The answer is, it's really in other cells there's problems too. But this is what he's presenting with. But we still need to talk about that. So let's talk about how we actually use the pentose phosphate pathway. So the whole goal of the pentose phosphate pathway is to produce certain types of molecules, whether they're the NADPH or you're producing ribose. So it all goes about how the cell needs ATP, how it needs NADPH, and how it needs nucleotides. So if you need nucleotides, you have, for example, rapidly dividing cells, you're producing a lot of DNA, you're producing a lot of RNA, you're going to need a lot more of that ribose phosphate. So you can go through the different phases of the pentose phosphate pathway. You go through your glucose 6-phosphate, and you can go through it, and you can follow the arrows through the different transketolases, transaldolases, and end up with a high concentration of ribose 5-phosphate, which you can then go and make your nucleotides. You'll end up with some more of the other end products, such as fructose 6-phosphate, or glyceraldehyde phosphate, but you can use them and shunt them back into ribose 5-phosphate and really concentrate that pool so you can get high levels of nucleotides that you need for these rapidly dividing cells. However, that doesn't happen on an everyday basis. So you're not always dividing. If you are, that's more of a cancerous type situation, and that's not very good. But on a daily basis, you could need NADPH much more than you need ribose 5-phosphate. So you can go through the pentose phosphate pathway from the glucose 6-phosphate, producing your ribose phosphate, and then cycle it back into fructose 6-phosphate and glyceraldehyde phosphate. But instead of shunting them into glycolysis, you can use these two sugars, put them back into the glucose 6-phosphate pool, and have them cycle around several times. And as they cycle around from the glucose 6-phosphate to the ribose to the fructose and the glyceraldehyde 6-phosphates, you're producing more and more NADPHs. So the whole idea is that we're recycling all these sugars around in this circle to create more and more NADPH so that you can end up with a high yield of this for your cells if you need that much NADPH. And there are certain conditions, certain synthetic reactions, certain tissues which really need high levels of NADPH. And so in those cells, those tissues, those organs, we're going to be doing this cycling of the sugars over and over again with the pentose phosphate pathway to meet their metabolic needs. Now, there's also a condition where you need both NADPH, but you also need ATP. So in certain reactions, you need both, or it could be in a very high metabolic tissue, such as a muscle, where you need a lot of ATP. But you also need some NADPH as well. So what you start off with is your glucose 6-phosphate. You go into your ribose at the end of the first phase, and then you produce your fructose 6-phosphate and your glyceraldehyde phosphate, and then you just shunt them into glycolysis. And so you then will eventually end up with your pyruvate and your ATP. And of course, from pyruvate, you can go further down into oxphos and do all of the other metabolic processes that we've already learned about. So in this way, we're producing the NADPH, which we need as a coenzyme, but we're also producing the ATP that we need for some type of enzymatic process or from some type of energy demand that those cells need. So as you can see, with all these different modes of use, it all depends on what the needs of the cell is. Does the cell need nucleotides? Do the cell need NADPH? Do the cells need ATP? With that kind of decision, you can go down the different routes of the pentose phosphate pathway and get to different results all using the same pathways and the same enzymes very succinctly and very easily, just cycling back and cycling around, doing reversible reactions, doing several different types of reactions to try and get to your endpoint that you want. Yes? Not particularly. It's not as efficient to do that, actually. Usually when you break down your nucleotides, you kind of don't go the step to break them into individual components. You can break them down to like the AMP or whatever, A, G, C, or T, U, or whatever. But you usually will shunt them back into making DNA or RNA. It's just a little bit more metabolically more cost effective to do it that way, so that you have those precursors when you want to synthesize RNA or DNA all ready to go rather than go fresh. So the pathways that use NADPH and ATP, or just NADPH alone, are very important. And so, of course, we have these this pentose phosphate pathway to help regulate them and to help replenish the NADPH pool so that the pathways can continue. Now, some of the tissues with very active pentose phosphate pathways include adrenal gland, which is involved in steroid synthesis, the liver for fatty acid and cholesterol synthesis. This is something we'll be talking about in a couple classes. The testes, as well as the ovaries for steroids, adipose tissue for fatty acid synthesis, mammary gland also for fatty acid synthesis. But look here at the bottom, red blood cells for maintenance of reduced glutathione. So pentose phosphate pathway is very important. So possibly this could be the link between what was going on with our hemolytic anemia in our patient and the pentose phosphate pathway. So all of these pathways, whether it's steroid synthesis, fatty acid synthesis, or maintenance of reduced glutathione, whatever the hell that is all dependent on NADPH. Of course, we're going to talk about what it is in the next slides. So now we'll talk about how all of this is linked to oxidative stress. And the very first picture on the lecture slide was the little cartoon of the superhero beating up the oxidative radical. So just as a review, we've talked about this in terms of mitochondria. Oxidants, oxidative stress, reactive oxygen species come from a variety of sources. Some are endogenous from metabolism with the mitochondria, inefficient leakage from the electron transport chain, for example, or with inflammation. There are enzymes that produce ROS to actually kill pathogens, but we also get it from outside sources, environmental factors like UV light, ionizing radiation, cigarette or other types of smoking, as well as air pollution and other types of environmental agents. They can all cause DNA damage, which DNA damage would then lead to cancer. But they can also do things that are very damaging to lipids, called lipid peroxidation. So essentially, a free radical formed by reactive oxygen species can attack the lipids, such as in a biological membrane, like a cell membrane, and then it changes the membrane permeability. So if it's a mitochondrial membrane, it'll become a non-functional mitochondria. If it's a nerve, it could dampen the nerve conduction, or if it's any other cell, it could, if there's enough lipid peroxidation, it could actually cause the cell to burst open. So this lipid peroxidation will damage organelles. It can also further formation of lipid radicals go on and damage proteins, DNA and RNA, and it will signal for cell death given enough of this damage. So of course, it's very important that we have protection from this. And the main source of protection is glutathione. It's abbreviated GSH. It is a tripeptide. It's consistent of glutamate, cysteine, and glycine. The interesting thing, though, is that the glycine and the cysteine are held together through typical amino acid linkage through a peptide bond. However, the cysteine and the glutamate are not connected through a peptide bond. Rather, the amino terminus of the cysteine is attached to the R group of the glutamate. This makes it very different. You can't build off of this to make it even larger. It's kind of stuck in this position. And we have glutathione being this tripeptide. It's in millimolar quantities in our cell, which is very high concentration. And it will react with reactive oxygen species through the thiol group of the cysteine. And so when it does that, the glutathione becomes oxidized, which is, you can see it dimerizes on itself, forming this sulfur-sulfur diester bond. And this is called oxidized glutathione. And so if you have enough oxidative stress, you're going to use up a lot of your reduced glutathione and oxidize it. Now, in cells that have DNA, for example, it's very easy to bump up the production of glutathione by bumping up the enzymes that produce glutathione and synthesize it. However, in something like a red blood cell, which does not have a nucleus, it cannot actually synthesize the enzymes to create glutathione. And so when you use up all that reduced glutathione to make this oxidized glutathione for this during this Ross damage, something's got to give. If we can't resynthesize that GSH, you're going to have damaged cells. So there are pathways to regenerate the reduced glutathione from the oxidized glutathione, and it is directly linked to the pentose phosphate pathway. So there are enzymes which will change the GSSG, so our oxidized glutathione, into our reduced glutathione, done by this enzyme called glutathione reductase. Now, glutathione reductase needs NADPH. So when NADPH levels are low, you can't convert the GSSG, the oxidized glutathione, into the reduced glutathione, and then you would actually deplete it. So having a very active pentose phosphate pathway to replenish that NADPH pool will allow us to convert that GSSG back into GSH to help with that reactive oxygen species defense. So coming back to our patient, the fava beans themselves contains many different compounds, one of which is called visine, that create reactive oxygen species. This damages the cells, all cells. However, in the red blood cells, because they can't synthesize extra glutathione, the cells will eventually burst. The red blood cells rely exclusively on the pentose phosphate pathway to reduce GSSG into GSH. And they cannot replenish GSH through de novo synthesis, so they can't actually create glutathione on its own. Whatever glutathione they have when the red blood cells are born, it stays there. It just has to be converted between GSH and GSSG. Noah? That is the oxidized glutathione. So essentially, it's GSH bound to GSH. So you have the SS together and like that that thioester linkage. So people who carry the gene for favism are protected, however, from malaria. The idea is that protozoans that cause malaria require red blood cells for their infection and to reproduce. However, if you have these hemolytic red blood cells, it prevents the organism from spreading the infection. So they get infected with malaria, the red blood cells burst, the malaria protozoan can't reproduce, and so they're protected. And so, on that hand, having the favism is actually something beneficial, and it's kind of the idea of why that kind of gene has actually stayed around in the gene pool and not been weeded out. However, these people have to be very careful about what they eat and what chemicals they're exposed to, and also what drugs they take. So, other than fava beans, other known chemicals that are that trigger the hemolytic anemia include henna, naphthalene, so in mothballs, and drugs such as pyrmocine, sulfonamides, some analgesics, and some non-sulfa antibiotics. They can all trigger hemolytic anemia. So when these types of drugs are prescribed to patients, there is a little red flag that says you need to screen genetically for this deficiency, because if you don't, these people will go into hemolytic anemia and potentially die because of that. So it's a very important screening that has to take place when these drugs are in use. So to summarize, our pentose phosphate pathway produces NADPH and ribose 5-phosphate in the oxidative phase. You can use this to produce your nucleotides. NADPH is an important cofactor for many processes, such as ROS defense, producing that reduced glutathione when you have that oxidized glutathione. And the non-oxidative phase can be a link between that ribose 5-phosphate to enter glycolysis. And so it all depends on those energetic and cofactor needs of the cell. Any questions? Yes.
So hypothetically speaking, if you had a patient and they were going on one of these drugs, and you asked them if you have any food allergies, would it be possible that they would mistake their condition for a simple allergy to fava beans?
Not really. The type of responses that they get
It's just too
is too severe. It doesn't present as a food allergy.
There's no such thing as a mild presentation of this.
Women do get a mild version of it. However, they would still do the genetic screening. Women might think, they just get sick when they have it if they don't have a severe case of it. It is an X-linked condition, so when women having two X's, essentially one gets inactivated, but it's more of the disease one present versus the non-disease one. Heterozygous, for example, it could be more severe, more of the one is present, less of the disease one is present, if you have more of a normal phenotype. So there's a wide spectrum with women. With men, it's either you have it or you don't, and too bad if you don't.
What did you read
Yes. Sure.
The question 9 said that it converted pyruvate into something using pyruvate kinase, but on our slides it's saying that phosphoenol pyruvate is converted to pyruvate using pyruvate kinase.
Pyruvate's converted to acetyl
Go away.
With pyruvate kinase too?
Yeah.
I hope so.
Let me just pull up the slide. Let me just